from the last time, about homework, about tests coming up, so I was gonna say, yeah, right.
So for the questions in the paper, you say draw out the path, but you want us to just talk you through it, or
You know,
actually
if
have
you
a
can,
picture?
oh, so if you can talk me through it, I'm okay with it. If you draw a picture and take a J, you know, if you draw a picture, take a picture of it and insert the JPEG on your paper, that's okay. The worst case scenario, and you probably won't do it, but the worst case scenario is you say, according, you know, look at the picture. You need to, whether you insert the picture or not, you need to walk me through the pathway, okay? Don't just say refer to picture and say nothing about it, because I know enough times people can draw the picture, but they don't know what it is that they've drawn. So you can do that. And obviously, that picture that I'm asking you to draw is probably going to be one of the cheat sheets that you bring in for the test. And this is the first year I've had homework due ahead of the test. In the past, it hasn't been a problem because the students have brought their homework to the test. So.
Is it going to be due at 5 on Friday or on Monday?
Monday, 5 on Monday. And I haven't opened up to this afternoon while I'm sitting here through the molecular genetics test. I will create the, don't say, okay? I'll open up the assignment place for Scholar. And I'll try to put it on at the very bottom of the Unit 2 stuff, because that is getting the stuff that I'm putting on is getting longer. Chow, you might want to move up. No.
Oh, okay.
There you go. Yeah. Sorry about that, guys. We're recording today. Okay. So, any other? So, I should ask. It's okay with leaving? Yeah. Well, I was going to say you could bring your homework to the test, but I don't think you're going to need it. So, but you can bring that drawing. That drawing should be very helpful to you on the test. Okay. Any other questions? We never did go through that one experiment that I gave you last week when I was at the Global Health Conference. I would like to do that eventually, and your interpretation of that particular experiment will be part of the test. Okay, so that'll be part of the material I can ask you about. So we can do that either at the end of today if we have time, or we can do it certainly next week. Any other questions? Yes.
So when you say cheat sheets, how many, like, do you want one paper front and back, or are we allowed to bring multiple pieces of paper, or how does that work?
You can do whatever you want as long as only protein names appear on the piece of paper. I mean, like, you can draw drawings and whatnot. There's, to have too few and too many, there's benefits to having too, you know, you gotta find a medium that works for you. If you have too few, you've probably not brought enough information, or they're somewhat incomplete. If you have too many, there's no way you're going to be able to sort through them during the test. Because I'm not gonna say, well, since you all have this, 500 pieces of paper you have to sort through, I'll give you an extra half hour. You're gonna have the same time frame. So you're going to have to decide for you what's complete, but not so onerous that I can't refer back to it. I'm not going to be going flipping pages all over the place. One person last year consistently brought her cheat sheets as one great big poster board. I'm not sure that was effective necessarily. And some people had just one or two cheat sheets and not complete in cheat sheets, so I had the range. Okay, if it was me, I'd put as much on a page as possible, as long as it was all related. Okay, because you do need to see connections and overlaps and all that kind of stuff. So I think for you to draw the connections, because it sometimes takes us more than one day to go through a pathway, so on your cheat sheet, you should be able to fuse those pathways together. So like Mondays and Tuesdays, get those pathways, I start on Monday, finish on Tuesday, get them fused together. And that's a good way to use cheat sheets.
So as long as it's just the protein name, it's
Yeah.
okay?
Yep.
All right.
Like I said, I had one person one time, this a long time ago, somebody put one protein on each page. And obviously she missed the connections between and pathways. So you don't want to do that. I mean, because there's really no advantage of having she had a giant circle on the page with the name of the protein inside the circle. I'm thinking, well, what good was that? So that was probably the most confusing. Like, what was that about? It wasn't, no, it was way before your time. Way before your time.
Way better.
Way back. You guys
Great,
were
understand better.
probably still eating in the lunchroom somewhere.
Those are the days.
Those were the days. Eat your food only. I said, that was the one.
We traded all the time.
ate people's food all the time.
I know, I was not, because I remember seeing in the rules in the lunchroom, I could get the keep your hands to yourself thing, but eat your food only. Like, which kid did?
Like, oh, you're not gonna eat that?
Yeah. Someone at the dinner last night didn't eat his tomatoes that we were on the salad night. I almost picked up my fork and went. Okay.
Get bags of chips into a mass pop.
All right, so we have a test next week. So next Wednesday, it'll start at 11:30 and you can have till 1:45 on it. Okay? All right, good. All right, well, I am ready to talk about kind of fuse movement through the Golgi and lysosomal proteins, kind of fuse those together in one lecture because we're sort of behind. Okay. So if you look on your picture of all of the different vesicular transports and different pathways, we're going to think about the Golgi for a minute. Okay. Oops. Oh. This, and these are medials. Okay, so there was at one time an argument. This is a long time ago. There was some sort of description of what the Golgi apparatus is, and people thought that the cis Golgi, notice that there's lots of vesicles coming into the cis Golgi all of the time. I mean, some are going back, but there's a lot of stuff coming into the cis Golgi all of the time. And so the idea a long time ago was, well, then then all of these fusing things at some point form a new cis Golgi, and then this cis Golgi gets pushed down to a medial Golgi, and then this Golgi pushes down to this Golgi, and then finally this Golgi gets pushed down to this Golgi. So that there was a continual birth of Golgi membranes up here, and a continual dissolution of membranes at the other end. So the idea was called cisternal progression. Okay, so that's what I probably probably taught when I first started teaching biology is that Golgi apparatus is made up here, and each stack gets pushed out. So this stack is older than this stack, and et cetera. Okay. Well, then people started using protein markers and discovered that proteins that are involved in events in the medial Golgi are always in the medial Golgi. And proteins that are involved in making some protein modifications in the cis Golgi are always in the cis Golgi. And proteins that do a job down here are always in the trans Golgi. So for a while, that was the whole idea of cisternal progression went away, because how can you have a distinct population of proteins in a medial Golgi that you don't find in the cis? And how can you have proteins that are unique to the trans Golgi you don't find in the cis Golgi? So the whole idea of cisternal progression died, and we thought, no, there is a distinct medial compartment, there is a distinct trans compartment, and medial Golgi never become trans Golgi. Okay, well that didn't last long. Okay, so now we're back to cisternal progression. Okay, so now we have to figure out, well then how can we have cisternal progression if there are medial Golgi residents that you don't find in the trans Golgi, and there's cis Golgi components that you don't find in the medial Golgi? How can you have distinct compartments if there's cisternal progression going on? Okay, so it was eventually discovered that proteins yes, this Golgi stack is going to become a medial stack at some point, okay? But in order to keep the cis proteins in the cis proteins, they have to be brought back. Okay, so if you look at your picture of where vesicles are forming and where they're going, you'll notice, aha, there is always backward, okay, or retrograde movement of proteins from the medial Golgi back to the cis, from the trans Golgi back to the medial Golgi. So that's going on all the time. And it looks like, is it COP1 coded? These are COP1 coded? Okay. That will select the cargo. The other thing I should tell you about the Golgi is that it appears, and I haven't read much since then, that all resident Golgi proteins are transmembrane. They're not, there aren't any luminal Golgi proteins. Okay? And what's unique about the Golgi proteins is that they have an unusually long transmembrane domain. So they're different from ER plasma, the plasma membrane proteins. They're different from resident ER proteins in that they have, in their transmembrane domain, it's particularly long. So S1P and S2P apparently have pretty long transmembrane domains. So that's on your, so if you're looking at your study guide at the different address labels, that's the address label for Golgi proteins. Okay. So today we're thinking about how do you get lysosomal proteins to the lysosomes, okay? And according to your map of movement, how do you think lysosomal proteins are getting to the lysosome?
Dr. Singer,
Yeah.
can you explain how the retrieval is done from the medial back to the cis?
Okay, so
What
so
exactly
this is a
happens?
this would be a COP1 coded vesicle, and so the COP1 protein is locating cargo to take back. I don't know in this case how that cargo is being selected. You know, what is the magical sequence for that for selection? Yeah. Kate.
So if cis becomes medial, medial becomes trans,
So does trans then become the endosomes? Or does
Uh,
trans always stay trans?
Or what else? A
Like-
vesicle that's bound for the cell membrane, or a storage granule
Okay.
for regulated secretion of a protein. So these things are budding off the trans-Golgi network to head off someplace else.
Okay.
Okay, and I think you'll notice that if you're going to the lysosome or early endosome, okay, note that there is a clathrin coated vesicle forming. Or you might not see that in other kinds of vesicle formations. Okay? So this thing can go either to an early endosome or a late endosome. Okay, but it looks like if something's going out to this plasma membrane, it does not go through a coated vesicle that's been discovered yet. So uncoated things go to the plasma membrane. The clathrin-coated vesicles forming on the trans-Golgi network are going to end up, their proteins are going to end up in the lysosome. Okay. I have one other thing I wanted. So anyway, that's all I want to say about this, the Golgi.
Are we gonna talk about the clathrin coating and everything?
I will have to. Because look where else do you see it involved? Oh, we're just looking in intracellular. The other place clathrin-coated vesicles are involved is in receptor-mediated endocytosis, which I want to cover sometime next week. Okay? But I think it's going to be fairly easy compared to everything we've gone through. Okay, I'm going to draw one more, one more giant diagram. The one that I gave you in color looks like movement of things from vesicle to vesicle, from one vesicle that's to organelle to vesicle to organelle. So it's all intracellular transport. Okay, all intracellular transport. But there is a in order to understand some diseases that we'll talk about, you need to understand some other kinds of transport, some relationship between the cell membrane and internal organelles. Okay, if you're going to do receptor-mediated endocytosis, almost always there is a receptor to pick something up. Okay, so the thing that's brought into the cell by receptor-mediated endocytosis is binding to a receptor.
Is that red clathrin?
That's clapping, yeah. It's matching this. I'm trying to be a little color coded here. Clathrin, like COP1 and COP2, is going to just be used for vesicle formation, and as soon as the vesicle is formed, it's going to dissolve. Okay, and the vesicle either fuses or becomes an early endosome. I think it's good. There's a fusion event that takes all of the stuff that got into the vesicle created by receptor-mediated endocytosis. Is going to end up in an early endosome.
What is the receptor taking out? Is that just a protein or?
It's something huge. And what that means is it could be a protein. So we will look at, some examples of proteins. So, your mother's milk contains antibodies that the mom produces. Okay, so that antibody has to be it has to be moved by endocytosis from the mother's milk, or from the mother's bloodstream into the milk. So that's done by receptor-mediated endocytosis. Viruses get into cells this way. The influenza virus gets in this way. Then it has to escape it has to get out of the endosome before it gets stuck and degraded in the lysosome. LDLs, okay, the unwanted cholesterol lipoproteins get picked up by receptor-mediated endocytosis that way. So notice that big things. Viruses are big, proteins are big, and the LDLs are really large also. Okay? So there are no transport proteins there are transporters that can pick up a protein, because the protein that's getting picked up is probably as big as the transporter itself. The virus would be bigger than the transporter itself. Okay? So this is big things. That blue represents big things. Okay? The early endosome, I put this really, really long curly Q on it. And sometimes it's hard to picture what's going on. Okay, but this stringy thing sometimes separates off the early endosome. This would be called a sorting endosome, or a sorting vesicle, or sorting endosome. It has bunches of names. Sorting. No, this is the sorting. Let's call it recycling. I want to be. And here's what's happening, and I'm mixing a little bit of receptor-mediated endocytosis in with lysosomal proteins, but that's because it's important. Okay, so a very typical event is that at this low pH, the protein or the whatever got picked up falls off the receptor, and the receptor gets recycled. And the thing that gets picked up, the blue thing that got picked up, whether it was a virus or an LDL or some other protein, ends up in a different pathway. Okay, and we'll take a look later next week about how does that separation happen. Well, pH, the low pH of the endosome causes the separation of the ligand, that blue thing, from the receptor. And what allows the receptor to get into a recycling compartment is probably surface area. So we'll talk about that separation. Okay, so this becomes a late endosome. The difference is, to some extent, what proteins you would find in the late endosome, but the functional difference is pH. This is even more acidic than the early endosome. And the late endosome becomes the lysosome. Okay, let's see. The trans-Golgi network, as you're seeing, I'm going to draw my picture a little bit different. Let's just have this be the trans-Golgi network. I'm not going to put all the bubbles in it. So T, G, N. This is the trans-Golgi stack, okay? Trans-Golgi network. Okay, can take things, really, stuff can go back and forth between here. stuff can go back and forth between here. You can send stuff to the early endosome. So there's a lot of interplay in these between the Golgi network, the trans-Golgi network, and this plasma membrane, and all of these endosomes and things like that. Okay, clathrin coating is these things would be clathrin coated. Okay. If you were just sending something out to the plasma membrane, not going through the recycling compartment, I'm making it one way. Okay, that doesn't need to be clathrin coated. So exchange between the trans-Golgi network and the endosomes is done by clathrin coated vesicles. Okay, so these are so interconnected and extremely complicated, and people have called, slightly different definitions for what's an early endosome, what's a late endosome, so you can look at a bunch of models, like including the one I gave you, but if you compared a bunch of models, it gets really foggy between recycling, or the recycling compartment, the early endosome and the late endosome. Things get pretty cloudy about what compartment is this, what compartment is that. There is another process going on in the cell called autophagy. Anybody hear about autophagy recently? How? Huh? Yeah, the Nobel Prize in science or medicine was won by a bunch of guys who learned a whole lot about autophagy, and trust me, there's still a whole lot to learn about autophagy. This is a process that you can't ignore anymore, and so I've tried to ignore it, but just can't do it. So autophagy, if there's an old worn out, this is a mitochondria, or a bunch of proteins that the cell is, doesn't need anymore. In autophagy, a membrane begins to form around the thing that they want to, that needs to be degraded. And the selection is rather complex. Okay? The source of the membrane, as far as I know, is still up for debate. I have not seen a good article that says, here's where the membrane comes from. It might come from ER. I don't, that's to me seems the most logical. Okay, but eventually, these two ends will grow, and this end will grow this way, and we'll end up with a double membrane bound autophagosome. Okay? And the autophagosome has to fuse with the lysosome, and then the contents of the autophagosome will get degraded. Okay, and we know for a fact that abnormal autophagy is going on in a lot of chronic diseases, and cancer is just one of them. Oh, and I'm wasting too much time. I gotta tell you one more story about autophagy before we go on to lysosomal proteins. So a lot of studies with mice have shown that mice that have either underconsumed calories or just consumed enough calories live longer than mice who can consume more calories than they need. And the epidemiology of humans also shows that people who underconsume calories or just enough calories tend to live longer and suffer from fewer chronic diseases. And it took forever to figure out what is the molecular mechanism of that. Just like it took a long time to figure out what is the molecular link between obesity and insulin resistance. So the molecular link has to do with autophagy. Oh, yeah, Ishaya.
So what else can the autophagy bind to apart from the Golgi?
Oh, the autophagy, these are just spontaneously forming membranes. Okay, there's nothing, they're floating, these are not connected to anything. I don't know where the membrane comes from. But all of a sudden, a membrane begins to form around the worn out mitochondria or some protein that seems to be unnecessary. There is some selection going on, so the autophagy doesn't randomly pick up stuff. Sometimes there's too much autophagy in cancer, sometimes there's not enough autophagy in cancer, depending, so both ways, abnormal autophagy is problematic. Right.
So the receptor-mediated endocytosis, is that not part of the TGN?
Oh, it can be.
So it can be and it can't be?
Yeah, in other words, if you notice that there can be, it probably isn't going to be, but I can't say it's exclusively, it excludes the Trans-Golgi network. So normal path is for the receptor to go back, comes in, goes back. Guess how long this takes? For the receptor to get picked up, end up in the early endosome, get recycled, and back to the cell membrane. This takes about 15 minutes. It's a very fast process. This usually gets degraded, but it's not impossible for a protein to get to the Trans-Golgi network.
So is the wall that you have there with the little buddy, what is that just a cell or what?
That?
Yeah, like
Yeah,
just,
you know how when COP2 distorts the membrane to
Yeah.
form, Clathrin distorts the membrane to form a vesicle.
so is that from the ER then?
No, this is outside the cell,
Oh, okay.
okay? Oh, okay, yeah, this is outside the cell. Sorry, was that confusing?
Yeah.
Oh, I'm sorry. This is inside the this is cytoplasm. Oh, okay, Ishaya.
So autophagy can bind, or not bind, wrap around the mitochondria.
Yes.
And you can also wrap around the cancer cells.
No, it can't wrap around the cancer cell, it's inside the cancer cell.
Okay, so.
It's inside the cancer cell. So from here to the end of the board is inside the cell, and this is outside the cell.
So once it wraps around the mitochondria of the cancer cell, then the cell dies.
No, this is happening in all cells. This is happening in all cells. If the mitochondria is not functional anymore, or you don't need it anymore, you can get rid of it by autophagy. Because this is the only way you can get rid of an organelle, is by autophagy. So some proteins are removed by the proteasome. This just represents another way to get rid of proteins, by autophagy. Amanda, are you gonna ask?
That was, you answered my question.
Okay, so the link between under-consuming calories and reduced incidence of chronic disease is that many proteins in the cell are being